Hi, everybody. Welcome to a New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am really excited, actually, as always, to be talking to the brilliant minds in science and in functional medicine and systems medicine and longevity science. And today I'm going to be chatting with Dr. Nichola Conlon. And Nichola is a molecular biologist by training. She's specializing in the study of aging as a biologically complex disorder. After years focused on the early stage drug discovery with a leading biotech firm, Dr. Nichola founded Nuchido Laboratories to deliver disruptive innovation in the field of anti-aging, rejuvenation, and health span, driven by her belief that cutting-edge science should not lie hidden. Go, Nichola. Dr. Nichola is on a mission to democratize science and has a skill for translating and presenting advanced scientific science in a way that helps to educate and support people to take care of themselves, to take control of their health. Along with other leading scientists at Nuchido, Dr. Nichola has identified the right combination of targets to restore cellular NAD production back to youthful levels, leading to the development of a second generation NAD booster called Nuchido Time Plus. Dr. Nichola, welcome to New Frontiers.
Hello, thank you so much for having me, Cara. I'm really excited to have a chat with you today.
So I'm excited because you're bringing really a great scientific background to longevity science, to the natural products industry, to continuing researching natural products and looking at things that are really important to us here in sort of functional longevity, if you will. But you are uh, bringing together many different areas that I'm particularly enthusiastic about, and you've launched this extraordinary NAD booster product that we're going to speak about today in careful detail. So you're just perfect for me to join me on my podcast, and I want to get into understanding you, your background, your company, kind of, maybe a little more detail, a little more blowout of what brought you here. So let's start with like a big overview picture, and then we'll move into the nitty gritty.
Yeah, of course. So I'm not a medic. I am a scientist, and my sort of area of speciality is the science behind why we age, but at a cellular level. So obviously everything that we're experiencing in terms of aging is really starting deep within inside ourselves, in our cells, and all the pathways and different compartments within there, although we tend to recognize aging on the outside. So my sort of background is to be really understanding what is going wrong at the cellular level to cause what we recognize as aging. And I've been really fortunate to have quite an interesting background in that I actually worked in drug development, but I was developing drugs to slow cellular aging. Now, to the average person, when you say you're developing drugs to slow aging, they're like, what? That's crazy. You can't do that. But it's a huge area now within drug development, because as you and I both know, age is your biggest risk factor for all of the major diseases that drugs companies are trying to treat. Cancer, heart disease, Alzheimer's, everything, your biggest risk factor is your age. So the idea is there a way that we can slow aging at a cellular level? And if we can, will that have an impact on actually reducing our risk of chronic disease, age-related chronic disease? And the idea is that we would be looking to extend our period of healthy lifespan, so extend our health span, so that we're not getting chronic age-related diseases as we get older. So I was fortunate enough to sort of enter that world as it was just beginning within the world of pharma. So this was back in around 2013, 2014. And it was a really interesting time because I spent a lot of time really understanding the biology at a cellular level behind aging, what were the best things to target, meeting all the key players within the aging and the longevity community. And then the ultimate goal was, how can we take all this knowledge and look at targets within our cells? But there was two main things that really bothered me in the world of pharma or drug development. The first was how long it takes. So, going from where I was in a lab, looking at the latest cutting edge science to then actually getting that science out to people that will actually benefit from it, you're looking at least 10 years, more like 15.
Right.
So that was just, you see this really amazing science and think this isn't going to get to anyone anytime soon. The second thing that really bothered me was part of my job. We would send a load of molecules for screening in the labs. We'd then get the data back and we'd literally, have a list of these molecules work exceptionally well to at the bottom of the list, these just don't work at all and everything in between. And part of my role was I had to look at which molecules were patentable. So which molecules could the drug company own? And therefore they would be the ones that they would take forward into development. And what I'd often see was that the things that worked really, really, really well were not drug molecules. They were things that were naturally found in foods. They were things that were approved as supplements. They were not what a company could patent and take forward. So these things would just sort of get pushed to one side. And then the things that would go into development were, half as good. And it was like, wow, they're actually going to put all this money into developing something that just works half as well. So I get it commercially, but ethically, I was like, this is crazy because I just saw that time and time again. So
it's just ridiculously interesting. I want to just take a minute and say that is so interesting. Yeah, I was going to ask you, were there any drugs in the pipeline that we might have heard about? But I think you've answered that. I'm sure that anything that you guys have discovered so far is still sort of hanging around in the lab, going through many, 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 many hoops, and not even close to being ready for prime time. Is that true?
Exactly that. So there was a situation that I could see where it was like, we have amazing science that could benefit people. And we have molecules that can do this, that are already known to be safe and well tolerated and have, hundreds of millions of exposures because they're in our food and things like that. And they're just not getting the research that they deserve. So I decided that I was going to leave that world. So I left in 2017 and I decided to found Nuchido Laboratories. And my whole mission was, how do we do something with those molecules? How do we get them the research and, all of the evidence base and the testing that they actually deserve? But I'm sure you can imagine what people said. They thought I was crazy when I was leaving my really credible job in drug development to start a supplement company.
Yeah. Oh my God, that had to be pretty hard. Was it? I mean, was it? You had some good support behind you, I hope. But yeah, I can only imagine the kind of
because obviously, supplements have a bad name. There's a lot of things out there that don't work as advertised and don't have much science behind them. But, from my perspective, I was like, the way that you identify molecules is exactly the same, whether you're looking for a drug or something that's not a drug. And actually, our bodies have no idea what the heck a drug is or a supplement or a nutrient. Like, our body doesn't go, oh, that's a drug, so it must work. And that's a nutrient, so it can't possibly do anything. Our bodies have no idea. They all have an equally, strong physiological effect. It's just that I wanted to bring the more credible side of testing and, the scientific credibility out of the drugs world, but bring it into the supplements world.
Very interesting. We saw this a lot in the COVID, the fervent COVID research happening, where there was a lot of exploration in molecules that might be effective before we had any grip on how to address this virus. And as often as not, they were all coming out in preprint at the time, but as often as not, it was a natural product that appeared to be as potent or more so than some of the, some of the drug molecules that they were exploring for efficacy with COVID. So very, very interesting. So you took this huge risk in leaving a credible, probably, an interesting career. I think you've explained it well, and it'll be interesting to a lot of our listeners, that you were seeing all these molecules that were fabulous. It's kind of cool to get a behind the scenes look from a scientist who's there, and these are natural molecules, so let's just sweep them off to the side. And I could see that would be an ethical tug for a lot of us listening. So you decided to make this leap, and what molecules grabbed you, and let's kind of move over to talking about that stage in your career, and what you've decided to focus on.
Yeah, I mean, I think one thing I did learn from drug development was that, again, I was fortunate to work for a very forward-thinking company that was investigating new drug development techniques. And they specialize in something called systems pharmacology. So conventional drug development basically will go, okay, we have a disease, we have a gene or a protein or an enzyme or something that goes wrong, and we'll just get a drug and we'll stick it to that particular pathway or protein or whatever, and that'll fix it. And as we know, 9 times out of 10, it doesn't, because biology is incredibly complex. And often there's not just one thing that's going on. And also molecules and drugs don't stick to just one thing in the body. They have footprints and they affect many different things. So I specialized in something called systems pharmacology, which actually was a more advanced discovery technique that took into account all of this. So it was basically, it addressed the fact that there was complexity. It addressed the fact that if you're going to have any real physiological effect in the body, that probably just doing one thing or looking at one target or one molecule probably wasn't going to cut it. And if you want to have real efficacy and real impact, then the best way is actually to look at multiple targets at the same time and use combinations of different molecules at the same time. And there you would get a much bigger effect. And the hit rate using that approach was way higher. So, not only did I want to look at how can we bring some of these, more natural molecules out of that world and test them, but actually is the way that you could be putting combinations of different molecules together. So they actually work in a slightly different way, or they're synergistic and have a more powerful effect. So a huge part of what we focused on at Nuchido Laboratories was, how are we going to take some area of aging science? How are we going to understand the physiology? And then how are we going to take combinations of molecules, use that systems approach to identify potent combinations of molecules that will then be regulated and approved as a supplement, so we can get it out to people very quickly, rather than waiting 10 to 15 years. And one of the first things that I really wanted to look at was NAD, because this had, back in 2017, had become a lot more popular. There was a lot more people talking about NAD within the aging space. And, for me, looking at all the research behind it, there was good promise. We now had a good idea of what was the underlying physiology surrounding it. So I really wanted to look at developing our first product as an NAD booster, but that worked better than the ones that were already out there.
Awesome. So, and you had did you have some kind of a notion on the natural products you were going to, really hone in on? I mean, you must have.
At the time, no, it was actually the targets. So we knew what targets that we were going to be looking at based on, the latest science around NAD. I mean, would it be helpful to do a bit of a NAD 101? Yeah. I mean, I'm sure everyone's familiar with it, but, so we know the reason, NAD it's just an incredibly important molecule in the body. And anyone that's done any sort of biology or medical training will have heard of it, because it's important for over 300 different reactions in the body as a coenzyme substrate.
I just want
And
to underscore that we're using NAD in different structures like all of the time. You know, I
yeah,
think in literally every cell, if I'm not mistaken, it's just
it's every cell. And
yeah.
I quite often say it's probably, if you haven't heard of NAD, it will be the most important molecule that you've never heard of, because if you didn't have it, you would literally be dead in 30 seconds. It's that critical for our physiology. And it's most famous for its role in energy production. So in the Krebs cycle, which occurs in our mitochondria, which literally takes the food that we eat and converts it into ATP, which is the energy that all our cells need to survive and do all of their functions. So incredibly important for that. The other thing that it's been becoming more famous for now is its role in cellular maintenance and repair. So NAD is now known to basically act as a bit of a signaling molecule in the cell, where it tells the cell to switch on and off maintenance and repair pathways such as DNA repair enzymes, the things that are going to make sure our cells stay in good health. So as a general rule of thumb, if you have high levels of NAD, you have good levels of energy production and good levels of cellular maintenance and repair. And if you have low NAD, energy goes down, maintenance and repair goes down. So the main reason why NAD can seem to regulate all these different functions is that it works in tandem with a group of proteins known as the sirtuins. Some people might have heard them referred to as longevity proteins. And this is because these sirtuins are known to actually switch on a whole host of downstream pathways and processes that are beneficial for cellular health. So that switch on recycling, switch on repair, switch on DNA repair. The sirtuins basically switch all of these things on. And the link with NAD is that they are absolutely critically dependent on NAD. So NAD basically acts as a fuel to power the sirtuins. So without NAD, they don't work.
What's interesting about that, just to kind of link it back to aging, is, I mean, you're going to do it anyway, but just off the top of my head, there was a paper that we've been interested in my group, the most recent Hallmarks of Aging paper. So this is teasing out all the mechanisms that really seem to drive aging, and NAD seems to have a finger in a lot of them. So they've expanded to 12 hallmarks. I think that originally there were 9, and this group has just recently expanded them. But NAD is right in the middle.
Yeah, it is. And that is one of the reasons why I chose, you know, of all the things we could have chose out well, at the time there was 9 hallmarks of aging, now there's obviously 12. You know, we have done a lot of research in areas like senescence, but for me, NAD, it was something that just everything seemed to come back to NAD. You know, if it was an NAD decline, it was linked to some of the hallmarks being established. If you restored NAD, it seemed to get rid of some of the hallmarks of aging. So it's very intertwined. And I guess it's not surprising because it's in every cell. It performs so many different functions that, you know, it's unsurprising that it's going to impact multiple hallmarks. But yeah, the reason that NAD is linked to aging is because it's been found to decline exponentially in our cells as we get older.
Why though? Why does it drop like a tank? Yeah.
So, well, it's estimated actually to drop by about 50% every 20 years. And that's from birth. In the tissues that are being studied, that's actually from birth. So, you know, you can see that even by the time you're 20, it's halved, then that halves again, then that halves again. So by the time we're in more advanced ages, you know, there's really not very much NAD left in our cells at all, which is pretty scary because it's so important to think that this molecule that we really rely on is becoming critically low. And for a long time, people didn't really understand what was happening to NAD. They didn't really understand the underlying mechanisms that were causing NAD decline. But it is now become well established what is causing this. But I think, first of all, a key thing to point out is that our cells are actually really good at making their own NAD. So when we're young, all of our NAD is actually just produced internally. So I think a lot of people think it comes from our diet or it comes from, some external source, but it doesn't. In young youthful cells, we actually have a pathway called the salvage pathway. And what this does is it basically recycles NAD continuously. So when NAD is used by cellular processes, such as the DNA repair enzymes or the sirtuins, they actually split it apart back down into some of its building blocks. And one of these building blocks is nicotinamide, vitamin B3. This nicotinamide is then actually recycled back into fresh NAD again. And that makes sense because NAD is so critically important that our cells almost have to have this fail-safe way to have a continuous supply. We wouldn't really want to rely on it coming from our diet, for example. So that's how it's produced in young cells. But we now know that the main reason why it's declining is actually this process becomes dysfunctional as we get older. So we start to see, I
want to ask you a question about that process, but I just want to say we had an interruption from my 5-year-old, my newly minted 5-year-old. She just turned 5, and she's an insane bundle of energy, right? Like you can see them, you can see NAD just busting out of the seams. Like it's interesting to think about it. And the more exhausted she gets, the more energy she gets, which is such a paradox. But it's funny that likely NAD is playing a big role in that. And by the time she's 20, she's gonna have a very different, and she's still gonna have a lot of energy, but it'll be different. So it's fascinating for me to think about that pearl that you just gave about NAD being so high at birth and then halving and halving and halving over time. Why is the salvage pathway breaking down as we age?
Yeah, so what we see is that there's a key enzyme within the salvage pathway. It's got a very long name, but we abbreviate it to NAMPT. So this NAMPT enzyme is what we would describe as a rate limiting enzyme. So out of the whole pathway, it's the really, really critical one, which its levels of activity are directly correlated to the amount of NAD that will be produced. And what we see in older cells is that NAMPT starts to get turned down. So this means that the key enzyme that's powering this recycling pathway just starts to go down with age. So it means our cells just simply can't make and recycle as much NAD as they did when they were young. So that's one key reason. But what we also now know is that the demand for NAD in older cells actually goes up as well. So right at a time when your cells actually need more NAD, you actually see a reduction in the ability of the cell to make it. And this causes a lot of other downstream knock-on effects, because what happens is, for example, we have a lot of inflammatory processes in older cells. There's a really famous one called CD38. It's an enzyme that seems to become upregulated with chronic low-grade inflammation as we get older. Now, CD38 just eats up NAD. It uses so much of it. And when it uses NAD, it gets broken down. And this is broken down into nicotinamide. Now, in young cells, this nicotinamide would just be flipped straight back into NAD again via the salvage pathway. But we know that enzyme isn't working as efficiently. Therefore, what happens in the cells is that older cells start to build up this nicotinamide because it's not getting recycled. And then the cell's like, oh my goodness, we need to maintain homeostasis. We don't like it when things build up. How do we get rid of it? And what it does is it increases expression of another enzyme called NNMT, which is a key methylation enzyme. And this enzyme sticks a methyl group on nicotinamide to signal its excretion from the cell. And then what you see is then further dysregulation, because you start getting methyl donor depletion because it's all being used up trying to get rid of this nicotinamide. And then you don't have the methyl groups to be involved in other critical processes like epigenetics.
Yeah.
So you can quickly see in older cells how you're having all of, you've got lack of recycling, you've got increased demand for NAD, you've got inflammatory processes using up NAD. suddenly everything's going out of whack. And, one negative thing leads to another. And then it's like the perfect storm for major NAD depletion. So I think the key takeaway is it's complicated. As with anything in biology, NAD biology is complex. And the reason it declines is complex. There's not just one thing causing it. There's multiple different things going on that cause the decline.
All right. And I know a lot of people are going to immediately be thinking about then, maybe B3 actually becomes sort of toxic, like to take. And I think you're going to address that in a little while, but, we prescribe nicotinamide in practice. We prescribe niacin, like a whole variety of those things. And if it can't be activated, then that may be a contraindication for use. And if it's not only not activated, but then it's methylated so that it can be eliminated, that could be another contraindication. My area of research has been in DNA methylation, and so we're always thinking about ways that we can preserve it. But and you're going to talk about that. But before you jump into that, I just wanted to ask you. This is a little bit of a wild card question, but I'm curious if you have anything to say about when we see this, the breakdown of the salvage pathway, the demand for it, the increased need as we age. So we need more NAD as we age, but we can't recycle it. It ends up accumulating. It's getting sucked up by CD38, and it's almost like aging looks almost like this program phenomena sometimes, like on purpose. We're just putting the body into dire straits so that it's vulnerable to, as you pointed out earlier, all the chronic diseases associated with, like all the big diseases are all associated with aging. And it seems like there's some sort of a program phenomena that's potentially occurring. And do you have any thoughts on that, or do you want to just kick that down?
Yeah, well, it's a hugely controversial area in the world of longevity. Is aging programmed or not? I think my take on it is that aging only ever makes sense when you think of it in terms of evolution. So, we've evolved to live to a particular age. We have evolved to live not long past childbearing age. So every process that we have in our cell is basically trying to protect us until we get to that point, and we've almost fulfilled our purpose, and then off we go. So why would evolution ever give us cellular abilities to live beyond that, to reach an age that we were never likely to naturally live? And this is why you have processes like senescence, which are highly beneficial when we're young because they prevent us from getting cancer. But then as we get older, they become hugely problematic. But in the reality, when evolution designed that process, we were never supposed to live until our 80s, and these cells weren't supposed to accumulate. So with aging, I always, say, if you're trying to explain anything in aging, just think of it from evolution's kind of point of view. But everything is so complex when you look at the hallmarks of aging. They're all highly interconnected. You can never look at anything in isolation. And this is the beauty of systems pharmacology. It really acknowledges that and doesn't ignore it, which unfortunately, a lot of scientists tend to have a bit of a reductionist approach where they're very focused on very particular areas of biology and fail to see the bigger picture of how their particular interesting area may be impacting something that they've never thought about before. So
it's great. It's just really cool that you have studied and have work in that systems model. It's just really refreshing. And indeed, I've interviewed people who are focused on single molecule activity and controlled activity, and it just misses so much. All right. And so that was a fun little tour through the importance of NAD and what happens as we age. Anything you want to add there, or do you want to circle back and start talking about, is there an issue with prescribing, nicotinamide or some of the, the precursors like nicotinamide riboside or NNMT?
I think a lot of people, well, I think first of all, going back through the sort of science of NAD. scientists seen this molecule, it's incredibly important. It declines with age. So why don't we just stop it declining with age? And if we did stop it declining with age, then would all of these important cellular processes remain switched on? Would we improve health span? Like how would it improve, you know, all the things that are associated with NAD decline? So scientists began to do things in cells and animals, then humans, that would boost in NAD. And lo and behold, found that if you maintained NAD or topped it back up, that you had a whole host of benefits. You know, obviously everything from increased mitochondrial function, energy production, physical energy, cognitive energy, reduction in neurodegenerative diseases, muscle, everything. It was like NAD seemed to just be, you know, this magic molecule that was addressing a lot of different issues when it came to aging. And again, it's like, it almost sounds too good to be true, but when you think about it, it's in every cell. It declines. It has all of these beneficial processes that, if they're getting switched off, is not good for cellular health. So, you know, it's no surprise that the benefits were so vast.
But
so people,
mostly in animals though, right? In animals,
yeah.
worms, you know, mice and worms, I think, but yeah.
Yeah, so you know, the early studies would have been your classic model organisms like your C. elegans worms, your Drosophila, etc. Then it was the mammals, and then more recently it's been the human studies. But there was a lot of, you know, how can we boost NAD? And it's known that NAD is quite a large and stable molecule. So the idea of just taking NAD as an oral pill was immediately, you know, wiped off the table. It's not possible. So the next sort of best idea was, well, why don't we use precursors to NAD? So like the raw material, the building blocks that our body uses to make NAD, why don't we take that?
Can you, Nichola, just speak to why NAD was wiped off the table? Just say that again. I didn't quite grab it, and I'm guessing some other people, like, why was it kicked out?
Yeah, so NAD by its very nature is an unstable molecule.
Okay.
So it's what we call a redox molecule, which means it flips between different states. So it loses and gains electrons and hydrogen all the time, and it flips really easily between these states. And that basically means that it's just very, very unstable. So if you try and put it in a pill or put it through your digestive system, it just gets degraded.
Okay.
The other thing is, let's say hypothetically, it did get intact into your body. Actually, it's a big molecule, and it's too big and charged to actually fit through our cell membranes. So there are not many cells in our body that have a transport protein that will actively intake it into the cells. So yes, you know, you could take NAD, but most of it would have to be broken down and then transported through as the precursors anyway. So they're the main reasons why NAD in a pill, you know, an oral pill was just not a practical solution. So the next best thing was, let's use the precursors, the raw materials. So these are what NAD is naturally made up of anyway. These are things like your niacin, your nicotinamide, your nicotinamide riboside, or NR, it's commonly known as, and your nicotinamide mononucleotide, or NMN. The most popular ones seem to be NR and NMN. And again, these are just precursors to NAD. They're the building blocks that our bodies use to make NAD. So the idea was, if we give the body and the cells more of the raw material, hopefully the body will convert it into NAD. And at the time it was so brilliant, you know, you can see it boosts NAD, and it's doing the job. But then this was in cells and model organisms. Then, you know, when it started moving through into humans, in the human clinical trials, there wasn't that amazing results with the likes of NR. And it was a bit puzzling. It's like, why have we had all this, you know, extending the lifespan of cells and health span and everything, and we're not really getting the effects translated into humans? So I think since those, you know, those the idea of using precursors came out, the science has obviously moved, and we now understand why NAD declines, as I've just explained the reasons. So in light of all of that, if you think now about what happens when you take a precursor such as NR or NMN, what we know is that it enters the salvage pathway. It actually enters lower than the rate limiting enzyme. So it does actually get converted into NAD, which is why in the studies you do see an NAD boost. But as soon as that NAD is being used at once via the sirtuins or the DNA repair or CD38 or whatever it is, it's broken down into nicotinamide. And then you've got the roadblock, because if they're older cells, then that nicotinamide and that pathway that's recycling it is not as functional as it should be. So essentially, yes, you're getting NAD, it's getting used once, but then you're getting a buildup of nicotinamide. And then you've got the problem of, oh my goodness, we've got to get rid of this nicotinamide. So the methyl donors start coming out, tagging the nicotinamide and excreting it. And this is why you'll see a lot of people talking about the fact that you should be taking methyl donors like trimethylglycine, choline, etc., when you are taking NR or NMN, because you can suffer from methyl donor depletion.
Let me ask you a question too, also.
Yeah.
So you've got this accumulation, this intercellular accumulation of nicotinamide. Does that have any feedback inhibition on the salvage pathway? So this salvage pathway that's already limping along as we age, is it further inhibited, or I mean,
so there's always a worry that nicotinamide can inhibit key things like the sirtuins. So, something that I always get asked is, oh my, why would you take nicotinamide? Because it inhibits the sirtuins. However, if you look at the data, actually in cells at ridiculously high concentrations that physiologically would never be possible, yes, it inhibits sirtuins, but in a physiological system, it doesn't inhibit sirtuins. It actually activates them via its role with NAD. So that's a bit of a myth that everybody seems to get a bit lost in, that it's an inhibitor. And again, this is because our bodies will do everything that they can to not let it get up to the concentrations where it becomes an inhibitor. So that's when you start seeing upregulation of NNMT to get rid of it. So in some of the studies that have been the human studies that have been done with NR, if you look at the data, what you will see is that as they increase the concentration of NR that they're given to the subjects, you can see a huge increase in the levels of methyl nicotinamide that are excreted in the urine, because again, that is not getting converted into NAD. It's not going around the salvage pathway. The body's suddenly having to upregulate a process of excretion because it simply can't deal with the amount that's coming in. So again, you're not addressing the root causes of the NAD decline. And by not doing that, you're actually causing another problem. Another thing to kind of think about and acknowledge, which is now people are now thinking about more, is that NAD that you're topping up actually going to the right place where you want it to be going? Because we want it to be activating the sirtuins, we want it to be activating the DNA repair enzymes. However, we know in older cells, they have increased expression of CD38, and CD38 has a much higher affinity for NAD than either the sirtuins or the DNA repair enzymes. So in a cell, what that means is that if CD38 is there enough, it's going to grab all of the NAD before any of the beneficial pathways actually have a chance to use it. So again, you could be thinking that by taking NMN or NR, you're actually boosting NAD and it's good because it's going to activate the sirtuins and repair. But actually all that NAD you're putting in is actually driving inflammation. If you don't look at addressing that other root cause of NAD decline.
That's really interesting. What cell type seems to house CD38? I mean,
a lot of the immune cells, but it's one of those that seems to have a bit more of a ubiquitous expression. But for us, this is why we talk about maybe second generation NAD boosting. Back then, we didn't understand all that. It was thought that, you know, using NR or NMN was the best way to approach the problem. We now know that actually it doesn't address the root causes and is potentially making some of the issues worse. So what we wanted to do was go, okay, we now know we've got an issue with NAMPT in the salvage pathway. We know that we have an issue with CD38. We know that we have an issue with methylation. What can we design in targets with system pharmacology that's going to target all of those different things and actually address the root causes of the decline rather than ignoring them, and address the complexity. So that's how we designed our formulation and our product to actually fix those issues.
So let's talk about it. Let's talk about what you figured out and how you put together this systems approach to dealing with that and what you found in your research. Let's get into it.
Yeah, well, you know, obviously from what we've discussed, one of our main targets that we wanted to affect was the salvage pathway. We know it's a leading cause of NAD decline. We know it's that key enzyme NAMPT that declines. So we wanted to boost levels of it. So for that, we basically use two different ingredients to do it that act on slightly different pathways. So rutin, it's derived from something called Sophora japonica. It has a high amount of natural quercetin in it. We know that that is an NAMPT activator. So that is a direct activator. The other ingredient is alpha lipoic acid. And this activates NAMPT, but via a slightly different pathway. So what alpha lipoic acid does is it actually activates an energy sensor in the cell called AMPK. And what AMPK does is it signals if the cell's in a bit of energy stress. And if it is in energy stress, what it does is it goes, oh, we need to switch on NAD production. So it sets off a series of reactions that basically lead to increased levels of NAMPT to increase NAD in the cell. And alpha lipoic acid actually activates AMPK. And the really interesting thing about that is it's that this exact pathway is activated by both exercise, calorie restriction, fasting, all things that are shown to promote cellular health and health span and lifespan in some cases. And it's known that a lot of the beneficial effects of these things are by increased NAD production. Because again, if you think about it, the reason our bodies are increasing NAD is that they've sensed this energy stress from the exercise or the fasting. Our body's setting off a reaction saying, we need to survive this stress. How do we do it? We increase NAD. Give the mitochondria more NAD to keep producing its own energy. We have NAD as a signal molecule to increase maintenance and repair and recycling so that we can get the cells through this period of no nutrients and energy stress. So yeah, that's a natural way of boosting levels of that enzyme, but obviously we're using a molecule to actually do that. And
very interesting. Yeah, keep going.
Yeah. ALA is a really interesting one. So, you know, it's something that you do come across frequently in the longevity space, as an antioxidant as well. And you've got to be really careful with ALA because in terms of what form that you use. So there's two different isomers. You've got an R and an S version. And quite often, if you look in products, they'll have the S, which is like the synthetic. And when you look at all the data, that does absolutely nothing in the body. It doesn't work at all. At best, you'll get a 50-50 mix of R and S forms. So again, you're kind of paying for half of it. That doesn't do anything. So I was really keen with our product that we only use our ALA. So it's only the form that we know actually works. And ALA is, it's one of these molecules that acts on many different pathways in the body. And another way that we know that it increases NAD is it actually activates a pathway or an enzyme called NQO1. And NQO1, what that does is it converts NADH to NAD+. So remember earlier, I said NAD is a redox molecule. Well, NADH and NAD+ are the reduced and the oxidized forms, and it flips between those constantly. But what you tend to find is that in older cells, it favors more towards NADH, and that's not good. You want it more towards NAD+. So what this enzyme does is it basically flips NADH to NAD plus. So you rebalance the more favorable ratio of NAD plus to NADH. So ALA acts as kind of a dual purpose. The other thing, again, that we wanted to fix was, if we've switched back on this NAMPT enzyme, so we know that the recycling pathway should be fully functional like a young cell, then what we want to do is prevent that methylation, because we want any nicotinamide to be getting promoted towards the recycling pathway rather than methylation and excretion.
Sure.
But we know in older cells, they've started to express more of this enzyme NNMT that methylates. So we use a green tea leaf extract, which has a high concentration of a molecule, which I'm sure you'll know, EGCG, and that is known to inhibit that enzyme. So what we want to do there is push the nicotinamide towards recycling rather than excretion. And the other interesting molecule that we use is parsley, which again, everyone's like, parsley, like why? But the reason we use parsley is because it's got a very high concentration of an active molecule that continuously came up in drug development called apigenin. People in the longevity world will probably know apigenin is a senolytic molecule. But for us, what we're using it as is to inhibit CD38, because it's been shown that even just inhibiting CD38 a tiny bit, you can have a huge impact on cellular NAD levels because it wastes so much NAD. So again, all things that are just trying to restore the balance of the cell back to like a youthful profile, like what it was doing when it was younger. Because at the end of the day, our cells are very good at making and recycling NAD when they're younger. It just all goes a bit out of whack. So that's, we also have nicotinamide in there as a precursor alongside these ingredients. And lots of people say, well, why would you not use NR or NMN? And the reason is because NR and NMN cannot freely diffuse through cell membranes. So again, they rely on transport proteins to actively take them into the cells. So they will only go in cells that have those transporter proteins, whereas nicotinamide is, it's uncharged, it freely diffuses through all cell membranes. And also we know, because of the salvage pathway, that it's the body's preferred precursor for NAD synthesis.
Fascinating.
So we have that in there.
But you're giving it in combination with all of these nutrients to help stimulate, the various pathways, so it's not just hanging around accumulating.
Exactly. So we would always say we would never give a precursor alone, especially in an older cell. Like,
right.
When you're trying to boost NAD, you really need to be given a precursor in combination with the other things to make sure that the precursor is going the way you want it to go and the NADs being used in that way. So back when I started the company, this was all the research we did. And then we were like, right, well, we've got to test it. Because, I was like, no, this, whatever we do, it has to be very well scientifically thought out, but also proven and tested, which is the key part that's missing with a lot of supplements. A lot of supplements make a lot of claims, but don't have any evidence in humans to back it up. And the benefit of using molecules that are known and tolerated and GRAS recognized is that you can put them into humans. You don't have to go through the really long drug development routes. So we initially did a pilot study, and it was just with 2 human volunteers, but we wanted to check, is it increasing NAD? Is it acting on the pathways that we wanted it to activate? And we confirmed this. So that was back in 2019. And then I was like, okay, so we know we have a formulation that works. It's stable. Let's do a bigger clinical trial. So we started a double-blinded, placebo-controlled crossover study, which in the world of trials is a gold standard trial in terms of design. We had 28 people enrolled. The people that we were enrolled were between age 20 to 80, and they were also male and female, which is really unusual because obviously a lot of clinical trials, you have to only use a very specific age group and usually men, just to try and eliminate any other factors. But for me, I was like, I want this trial to be as representative as the real world, because this supplement is going to go into the real world. So we need to know it works in a normal population of people. So we basically started that trial. Then obviously COVID hit, so we had to stop it. Restarted. So we wanted the data out for this, years ago. But we've actually now just finished, and it's unblinded. And what we measured in the study was, first of all, we wanted to see what's it doing to NAD. So we found it did significantly increase NAD, thank goodness.
Yeah, right.
Yeah, we then looked at NAMPT, and again, in all of these volunteers, we saw that NAMPT protein was upregulated. It went from barely detectable to actually being detectable in the cells of these people. We looked at the sirtuins and saw that the SIRT1, probably the most famous sirtuin when it comes to longevity, was it had significantly increased expression as well. We then wanted to look at, okay, so we know the NAD is gone up, we know the mechanism of action, like what are some biomarkers of aging that should be changing? So one of them that we looked at was levels of inflammation. Because, we know that chronic low-grade inflammation is associated with aging. We know that if we're inhibiting CD38, we should be reducing inflammatory factors. Let's see what happens. So we found a significant reduction in 4 different inflammatory cytokines. And then obviously that, is one inflammation. Chronic inflammation is one of the new hallmarks of aging. So for us, that was exciting. And that, inflammation is also something that can be measured in a clinic, unlike sirtuins or NAMPT, which I think is important. We also looked at levels of glycation. So glycation is, when sugar molecules start to become stuck to proteins in the cells irreversibly. It leads to a lot of problems like stiffening of the arteries, the skin, and it's a good biomarker of aging as well. And again, we saw a significant reduction in glycation. What else do we measure?
You looked at biological age. Something we talk about a lot in minor detail. Yeah.
How can I forget that?
How did you look at biological age?
So biological age was something we put in there because, again, this is a consumer product. This is, something that we want people to be able to understand. And we think looking at biological age is a really easy way of understanding something scientific. It's a number. You want it to go backwards. You don't want it to be higher than your chronological age. And that's kind of all you need to know. So we actually looked at glycan age. So we didn't do epigenetic markers, we looked at glycan age. So everybody that was at a trial did the glycan age test before and after each arm of the study. And again, we found a significant trend towards a reduction in biological age. After only a month of taking the study, it was about 1.26 years decrease. So I think we want to look at that in larger populations of people now, but for us it's a good start. It's a lot more we want to do a longer study that's more than a month, but to see all of those parameters changing in just 28 days, for us, we were really pleased that it's something it's something that is available. It's not a drug. But it actually could have some significant benefit, and it's got data to support it.
And I know this is finished the peer review process and it's heading towards publication shortly.
Yes.
We're going to link to all your papers and any sort of primers that you want the audience to have access to. We'll put them in our show notes. This is going to be a great paper to check out. I can't wait. Congratulations. Yeah, really. Congratulations on all that. You put together something that's moving the needle.
Thank you. No, it's been an interesting journey. Naively, when you start, you think, oh, how hard can it be? It's not a drug. It can't be any worse than a drug. But all the things you have to jump through, and then but I'm really proud of the team and what we've achieved, and managed to fulfill our mission of taking something from a lab and getting it into the hands of an everyday person, and being accessible, and being able to benefit people.
I would love to look at how this influences DNA methylation and, gene expression through an epigenetic lens, because, your product is packed with what we call epinutrients, that have
Yeah.
evidence in the literature for influencing specifically DNA methylation. Not just that, the enzyme, as you mentioned earlier, but um, and I've really come to appreciate the combination of nutrients like you would get in food. It's that combination of information, given at a single time that really seems to direct the symphony of information, if you will, to optimize cellular function. So I appreciate your product design quite a bit.
Yeah, and I think as well, some of the forms that we put the ingredients in, so like using rutin rather than quercetin and parsley for the
Yeah,
apigenin, that in those forms that you can have like the glycosides and they're protected slightly going through the gut, and they get cleaved.
fascinating.
And so my PhD was actually all in bioavailability. So I was always
Interesting.
incredibly passionate about putting things in the form that they're most likely to get absorbed and in the amounts that you need to get the oral bioavailability. So yeah, sometimes people say, well, why wouldn't you just use apigenin or just use quercetin? Why is it in different forms? But again, there's a well thought out reason for that as well.
Another pearl. I'm glad that you wrote that in. Yeah, yeah, it caught me earlier when you had mentioned it, the rutin, for quercetin. But yeah, thanks for fleshing that out. That's really fascinating. What about, like, you had NAD? I know actually a lot of my colleagues are offering NAD IV infusions in practice, or they're getting them themselves. I mean, it's just another huge route of delivery. And any thoughts on that?
Yeah. So I think I really wish there were more studies on NAD IVs to, clinically show what's happening when people are having them. I think there's a real lack of data and studies out there, but we know mostly anecdotally that people do report a big benefit from them, especially in areas of like cognitive decline and addiction and things like that. So I think, as I mentioned earlier, NAD is a big molecule. We know that it doesn't get into every cell. We know there are certain cells it can be actively transported into, like neurons, heart cells. So I think in those cells, when NAD does go into them, you've kind of got a little bit of a similar problem as what you have with the precursors, because in older cells or cells that have some sort of dysfunction, that means that their salvage pathway is not working properly, or they've got more inflammation or things like that. Again, you're delivering like a huge hit of NAD, and that NAD is going to get like a first pass, if you want to call it that. It's going to get broken down and used. And then you've got this massive influx of nicotinamide that the body suddenly has to deal with. So again, some people report when they're having these IVs that they feel a bit sick and they have palpitations. And I think a lot of that probably is this big influx of nicotinamide that the body's suddenly trying to methylate and upregulate and excrete and deal with it. So actually what we've started working with a couple of our practitioners that we work with who do IVs is saying, you know what, theoretically, if you combine our product with an IV and give the product for a month before you give the IV, it's almost priming the cells ready to be in a better situation to be able to deal with that sudden large hit of NAD that it's going to get. Because if you think about it, if you're upregulating your salvage pathway, and it means that when you are getting that huge dose of NAD, it gets used, it gets broken down, but again gets recycled, and it can be used again and broken down and recycled, as opposed to you have it once and then the cell can't do anything with it and it just gets rid of it. And you've just had a very expensive IV and it's kind of had a one hit and then it's gone. So, we want to do a trial on this, but anecdotal evidence shows that people seem to have a more prolonged benefit when they've almost primed their cells beforehand. And again, theoretically, it all makes sense. We want to test it, but anything you can do to make sure your cells are optimally placed to deal with the large dose of NAD is always a good thing. So again, you don't want to be putting a huge NAD IV infusion into massively inflamed cells with CD38 that are just going to take all of the NAD to the wrong processes, you know, and not the beneficial ones. So that's something we're looking at testing a bit more, but certainly, you know, anecdotally, we've had some good feedback from that.
Awesome. Yeah, it makes sense mechanistically, and it'll be interesting
Yeah.
to see what you find since you know, since you're looking at it. You had said before, and I just want to kind of underscore it again, that using your product is almost it could be like a fasting mimetic. It's actually upregulating some of the pathways that that fasting does and exercise does, as you mentioned earlier. But you might use it concurrently if you're doing a fast or if you're in a time restricted eating structure.
Yeah. So, you know, a lot of our customers say, you know, I practice fasting or time restricted eating. Like, when should I be taking this? So what we say is take it with your first meal, because we know that fasting is obviously increasing your NAD levels because it's activating AMPK. Then usually when you're eating your first meal, you know that NAD then drops again because that energy stress has been relieved. So by taking like a supplement with those mimetics in that are actually still triggering AMPK, you're actually prolonging the activation of those pathways, even though you've begun to eat. So in that way, effectively, you're prolonging the activation of the pathways that you've been activating for the last 16 hours fasting, even when you start to refeed.
And certainly there's plenty of people who don't have an appetite, no pun intended, for doing any kind of structured food restriction. You know,
Yeah.
it can be really triggering. We see it in our clinical practice actually with some frequency. So this might be an alternative to getting some of those benefits.
Yeah, absolutely. Absolutely. You know, that's not for everyone. But we know that, you know, lifestyle ways to boost NAD, it's exercise, it's, you know, it's clinically proven in clinical trials that, you know, lifting weights and doing high intensity interval training will boost NAD, calorie restriction, intermittent fasting. But again, not everyone is able to do that, or not everyone wants to do that. So yeah, this is another way of actually activating those pathways.
So just in our final, I have one more question for you, and then we'll kind of wrap up here. We're wanting to test NAD levels in clinical practice. I think there's kits offered to consumers these days. Are these labs ready for prime time?
So as of yet, I haven't come across any lab that's been able to offer a test that I would say is robust enough. And the reason is, you know, we'd love to be able to offer NAD tests and kits to our customers because we know it works, and, you know, being able to measure it yourself just seals the deal. However, NAD, as I mentioned, is incredibly unstable. And that means that it breaks down really quick. So obviously, if you wanted to just do a finger prick test and send it off in the post, by the time it gets to the lab, I can guarantee there will be no NAD left in there. You know, when we were doing our testing and our trials, etc., we actually did an experiment to look at stability of NAD in blood, just, you know, at room temperature, then on ice and things like that. And, you know, if you just left it on the bench, it pretty much gone in 20 minutes. So when analyzing it in the labs, you know, it would be intravenous blood draw, it would be straight on ice, and then it would be straight to be prepped like immediately to extract the cells and cryopreserve. So you were preserving any of that NAD that was there. So I haven't come across a company, although there are companies offering NAD testing, I haven't come across anyone that's convinced me that they found a way to preserve it. And this is why we went with, you know, for a consumer kit, a biological age kit, because, you know, there's good data on that. It could be a finger prick blood test. It's, you know, it's stable, it can be sent away. And it's things that customers and practitioners can actually do themselves for themselves or the patients.
Right. And also you mentioned, you know, inflammation. We can easily measure
inflammation
in practice. Yeah,
Yeah.
for sure. Well, Dr. Nicole, it's just fabulous to get to talk to you. Really interesting conversation. I'm excited about your new product. I'm excited about the research, the effort, the time, the energy, the money that you're investing into kind of teasing out what it's doing in humans. Again, everybody will link to the show notes, all the papers. that we covered today, so you have access. And as soon as the human study is out, the most recent one that will be coming out shortly, we'll get that up on the show notes. Thank you so much for coming, and I look forward to continuing the conversation with you.
Yeah, no, amazing. I've really enjoyed that chat. So thanks very much for having me.
My pleasure.